What if we didn't search for alien life? What if we accidentally smelled it? 700 trillion miles away, a distant planet is leaking a gas that, on Earth, only life produces. Dimethyl sulfide, biological waste. In December 2024, the James Webb Space Telescope detected that exact molecule drifting through the atmosphere of K2-18b, a world 120 light-years away. The confidence level is 99.7%. This is not theory. This is detection. K2-18b orbits a red dwarf star in the constellation Leo. Red dwarfs are the most common stars in the galaxy, smaller and cooler than our Sun, but extraordinarily long-lived. They burn their fuel slowly, capable of shining for tens of billions of years. This particular star is young by cosmic standards, only a few billion years old, and K2-18b completes one orbit every 33 days. That is its year. 33 Earth days to circle its sun. The planet sits in the habitable zone, the narrow band where temperatures allow liquid water to exist. Not so close that everything boils away. Not so far that everything freezes solid. Right in the middle, where chemistry can slow down and complexity can begin. But K2-18b is not Earth, not even close. It has 8.5 times the mass of our world. Picture Earth as a basketball you can hold in your hands. K2-18b would be a beach ball you would struggle to wrap your arms around. That extra mass means stronger gravity. If you weigh 150 pounds on Earth, you would weigh roughly 450 pounds on K2-18b. Every step would feel heavy, like wading through deep water. Your muscles would strain, your bones would ache under the constant pull. The atmosphere is nothing like the air you are breathing now. Earth's atmosphere is a delicate mix of nitrogen and oxygen. K2-18b's is dominated by hydrogen, thick, dense, pressing down on everything below. You could not breathe it for even a moment, but the strangest part of this world lies beneath that alien sky. Water, not scattered oceans broken by continents, not polar ice caps and exposed land. As far as current observations suggest, the entire planet is covered by a single, unbroken ocean. No mountains rising above the surface, no islands, no beaches, just liquid water in every direction, potentially hundreds of miles deep. Scientists call worlds like this Hycean planets, hydrogen atmospheres over global oceans. Until recently, we assumed habitable planets had to resemble Earth. K2-18b suggests that assumption was far too narrow. Different structure, different atmosphere, same essential ingredient. Liquid water. That ocean would feel alien. On Earth, the deepest trenches reach about 7 miles. On K2-18b, with stronger gravity and far deeper seas, pressure at the bottom could be immense, enough to force water into exotic high-pressure ice phases while remaining warm, states of matter that barely exist on our planet. And yet, somewhere within that vast ocean, or near the boundary between water and atmosphere, something appears to be producing dimethyl sulfide. On Earth, this molecule comes from life. Phytoplankton, organisms smaller than a grain of sand, release it as waste when they metabolize sulfur. It is not intentional. It is simply a byproduct of being alive. The gas drifts upward into the atmosphere, where instruments can detect it. If the same molecule is present on K2-18b, then life on two radically different worlds may be following the same chemical rules. The same pathways, the same signatures. Chemistry does not change from star to star. The same elements behave the same way everywhere. Given the right conditions, similar outcomes emerge. Temperatures on K2-18b appear moderate, somewhere between 10 and 50 degrees Celsius, warm but not hostile. The planet orbits closer to its star to compensate for the star's lower energy output, completing a year in just 33 days. The result is a world where liquid water can persist, and extreme temperature swings are unlikely. Picture the surface of that ocean. Endless dark water beneath a hydrogen sky. No landmarks, no coastlines, just the curve of the horizon in every direction. The sky would not be blue, but tinted red or orange by the dwarf star's light. The sun would appear larger in the sky, closer, yet dimmer, casting a muted glow across the waves. If life exists there, it lives under conditions we have never observed directly. Stronger gravity shaping movement, a hydrogen-rich atmosphere changing chemistry. Perhaps life never leaves the water. Perhaps it has adapted in ways we cannot yet imagine. For decades, we searched for Earth-like planets. Rocky, familiar, comfortable. K2-18b is none of those things. And yet, it carries a chemical signature that, on Earth, means life. The universe does not care about our expectations. It builds habitable worlds in whatever forms physics and chemistry allow. And sometimes, across unimaginable distances, those worlds leave behind traces, drifting quietly through alien skies. The question of how life might have started on K2-18b is a fascinating one. On Earth, the origin of life remains one of the great unsolved mysteries. We know it happened. Fossil evidence reaches back nearly 4 billion years. But exactly how chemistry crossed the threshold into biology is still unclear. How non-living molecules organized into self-replicating systems remains an open question. Multiple theories exist. Life may have begun in shallow pools where evaporation concentrated organic molecules, or in deep-sea hydrothermal vents, rich in chemical energy, or even in space, with organic compounds forming on comets and arriving during periods of heavy bombardment. K2-18b would allow some of these possibilities and rule out others. There are no shallow pools on a planet with no land, but hydrothermal vents could exist, and like Earth, the planet would have experienced early bombardment by comets and asteroids, potentially delivering organic molecules from space. The hydrogen atmosphere itself may have helped. Early Earth likely contained more hydrogen than it does today, and some origin of life theories suggest hydrogen-rich conditions favor certain prebiotic reactions. K2-18b has retained its hydrogen envelope, possibly creating a stable environment where those reactions could proceed. Time matters too. The star K2-18b orbits is a few billion years old, and the planet itself is likely similar in age. On Earth, life appeared within the first billion years. Once established, it had billions more to evolve. K2-18b has had comparable time, enough for life to emerge, adapt to strong gravity, spread through a global ocean, and develop metabolism suited to a hydrogen-rich world. But caution is essential. The detection of dimethyl sulfide is not proof of life. It is evidence consistent with life. Scientists must ask whether non-biological processes could produce the same signal. Researchers have modeled geological and chemical pathways, testing whether sulfur compounds could generate dimethyl sulfide without biology. So far, no convincing alternative explains the observations. That does not mean one cannot exist. It means that, with current knowledge, life remains the best explanation. The implications extend far beyond a single planet. K2-18b is not rare. It orbits a red dwarf, the most common type of star in the galaxy. Roughly 70% of Milky Way stars are red dwarfs, and planets in their habitable zones are common. Thousands have already been identified. If one of them hosts life, then life may not be an extraordinary accident. It may be a repeatable process, chemistry becoming biology, wherever conditions allow. This is the shift K2-18b represents. We move from asking whether life exists elsewhere to asking how common it is. From wondering if we are alone to realizing we may simply be one example among many. Not the living world, but one of countless worlds where life found a way. But how do we actually know any of this? How can we be certain that what we are detecting is real and not an error in the data, not a misreading of signals that have traveled for more than a century through empty space? The answer lies in a technique so elegant it borders on the miraculous. We are reading the composition of an atmosphere on a world we will never visit by watching how starlight changes as it passes through that atmosphere. We are not sending probes. We are not launching spacecraft. We are using light itself as our messenger. and the message is written in the language of missing colors. The method is called transit spectroscopy. It works because every molecule absorbs specific wavelengths of light. Water vapor absorbs certain infrared wavelengths. Carbon dioxide absorbs others. Methane absorbs others still. Each molecule has its own pattern, as distinctive as a fingerprint. This is not theory. This is physics we test in laboratories on Earth by shining light through known gases and measuring exactly which wavelengths disappear. Now scale that up to planetary distances. K2-18b orbits its red dwarf star every 33 days. From Earth, once per orbit, the planet passes directly in front of its star. This is a transit. When it happens, the star's light dims by a tiny fraction, far too subtle for human eyes, but measurable by sensitive instruments. Here is where it becomes remarkable. Not all of the starlight is blocked. Some of it passes through the thin shell of atmosphere around the planet's edge. As that light filters through hydrogen, water vapor, dimethyl sulfide, and other gases, certain wavelengths are absorbed. The light that reaches us is missing those colors, like a rainbow with specific bands cut out. Those gaps tell us exactly what molecules the light encountered. Think about this in practical terms. A photon leaves K2-18b's star and grazes the edge of the planet's atmosphere. If its wavelength matches the absorption signature of dimethyl sulfide, the molecule captures it. That photon disappears, absorbed 120 light-years away, in the year 1904. Other photons pass through unaffected and continue their journey, traveling for 120 years until they reach our telescopes in 2024. The precision required is extraordinary. K2-18b is 120 light-years away. The planet is a speck passing in front of another speck. The light passing through its atmosphere is a tiny fraction of the star's total output, and we are measuring changes in that already faint signal at the level of individual wavelengths. It is like identifying what someone ate for breakfast by analyzing a single molecule of air they exhaled after that air has traveled 1,000 miles. But it works. The James Webb Space Telescope observed multiple transits of K2-18b. Its instruments split the starlight into thousands of wavelengths and measured each one precisely. Most appeared exactly as expected, but specific wavelengths were dimmer. Those dim bands matched, exactly, the laboratory absorption signature of dimethyl sulfide. Not approximately, not loosely, precisely. The signal repeats. Different transits show the same missing wavelengths. Multiple instruments confirm the pattern. This is not a one-time anomaly or a calibration error. It is a repeatable detection that strengthens with every observation. As the data accumulates, the probability of error drops. Eventually, it reaches that 99.7% confidence level. The light carrying this information left the system in 1904. That photon began its journey when your great-great-grandparents were young. It traveled at 186,000 miles per second for 120 years, crossing distances that make the solar system look microscopic. K2-18b is 700 trillion miles away, roughly 7.5 million times the distance between Earth and the Sunday. Along the way, the photon crossed interstellar space, so empty it defies intuition. Between stars, there may be 1 atom per cubic centimeter. The air you are breathing contains millions of trillions. For 12 decades, that photon traveled through near-perfect vacuum, encountering almost nothing at all. It could have been lost, scattered by dust, bent by gravity, altered by charged particles. But this one survived. It arrived at exactly the right place, at exactly the right time, and struck JWST's mirror. There, its journey ended. The light was split, its energy measured, its data processed, and scientists read the message it carried. The unmistakable absence of specific wavelengths, the spectral fingerprint of dimethyl sulfide, 1 photon among trillions, but enough to tell us that something, somewhere, changed that light. Now think about what is happening right now on K2-18b. At this very moment, new photons are leaving the planet's star. Some of them skim the edge of the planet's atmosphere, passing through that thin shell of gas. Dimethyl sulfide absorbs certain wavelengths, water vapor absorbs others. The surviving photons begin their 120-year journey toward Earth. If humanity is still around in 2144, someone will capture those photons and read the information they carry. They will see K2-18b as it exists right now, in our present moment, and whatever happens next on that planet, we will not see it for another 120 years. After that, forever observing the past, forever delayed by the speed of light. This creates a strange temporal relationship between our world and K2-18b. If life exists there, if there are organisms advanced enough to wonder about the universe and build telescopes of their own, they would be seeing Earth as it was in 1904, a planet with no satellites, no global radio noise, no city lights bright enough to register across interstellar distances. They would see a living world, but no visible civilization, and we see them the same way, delayed by the same span of time. If intelligent life exists on K2-18b, we would not know. Intelligence is invisible at these distances. Technology is invisible. All we can detect is chemistry, the bulk composition of atmospheres, the presence or absence of specific molecules. Light can reveal chemicals, but not consciousness. Transit spectroscopy only works under precise conditions. The planet must pass directly between us and its star. The orbital plane must align almost perfectly with our line of sight. Most planetary systems are not oriented this way. Most planets will never transit from Earth's perspective. We are fortunate that K2-18b does. That accident of geometry makes these observations possible. Each transit lasts only a few hours. The planet takes 33 days to complete an orbit, but the window when its atmosphere is backlit by the star is brief. During that time, telescopes must collect as much light as possible. Miss the window, and you wait another 33 days. JWST observed multiple transits to build up signal strength. Each one added data. The first showed hints. The next added confirmation. After several transits, the signal became clear enough to identify individual molecules with confidence. Along with dimethyl sulfide, the spectrum revealed water vapor, expected for an ocean world, but still important confirmation. It tells us evaporation is active, that liquid water is present at the surface, cycling into the atmosphere and back again. Methane also appeared, though at lower concentrations. Methane can be biological or geological. By itself, it proves nothing. What matters is the combination. Carbon dioxide showed up in smaller amounts, as expected for a planet with a global ocean where CO2 dissolves into water and participates in chemical balance. Carbon is essential for building complex molecules, and its presence provides raw material life could use. Together, these gases form what astronomers call a biosignature ensemble. No single molecule proves life, but certain combinations, especially gases that should not coexist for long without continuous replenishment, suggest active processes at work. On Earth, that process is biology. K2-18b lacks detectable oxygen, which makes sense in a hydrogen-rich atmosphere where oxygen would quickly react and vanish. But the presence of dimethyl sulfide alongside methane and carbon dioxide suggests an atmosphere held out of equilibrium by ongoing inputs from the surface. Life is the simplest explanation, though not the only conceivable one. Confidence comes from consistency. In laboratories on Earth, dimethyl sulfide absorbs infrared light at specific wavelengths. In the spectrum of K2-18b, absorption appears at those exact same wavelengths. The match is precise, too precise to be coincidence. A molecule is a molecule. Its structure determines how it interacts with light, no matter where it exists. This universality of physics is what makes astronomy possible. The same laws apply everywhere. Distance does not change them. Time does not change them. The photons arriving today from K2-18b were emitted 120 years ago, but they carry information shaped by the same molecular processes that would occur if we could sample that atmosphere directly right now. In effect, we have sampled it, not by traveling there, not by sending a probe, but by reading the information encoded in starlight. Every transit adds another measurement, another layer of understanding. Over time, we will build a detailed picture of that distant atmosphere, its composition, its stability, perhaps even its changes, all from 120 light-years away, all using nothing but light. The detection of dimethyl sulfide represents the culmination of centuries of scientific progress. Learning that light carries information, learning how molecules imprint themselves on that light, building instruments sensitive enough to detect signals this faint, step by step, generation by generation, until we reached a point where we could search for life, not by traveling outward, but by listening carefully to the light that reaches us. The James Webb Space Telescope that made this discovery represents 3 decades of human ambition, engineering, and stubborn refusal to accept impossible. The project began in the mid-1990s, when astronomers realized that the next generation of space telescopes would need to observe in infrared wavelengths to study the earliest galaxies and the atmospheres of distant planets. What started as a concept grew into the most complex scientific instrument ever launched into space. $10 billion, 30 years of development, thousands of engineers and scientists, and a launch day that felt like holding your breath for humanity's future. The core challenge was size. To collect enough light from distant, faint objects, the telescope needed a mirror much larger than any previous space telescope. The Hubble Space Telescope, launched in 1990, has a mirror about 8 feet across. It's done incredible work, transformed our understanding of the universe, discovered things we didn't know existed. But its mirror is too small for the kind of observations JWST was designed to make. To see the faint infrared glow from the atmospheres of distant planets, to detect individual molecules in those atmospheres, you need more collecting area, more mirror surface gathering photons. The engineers settled on a mirror over 21 feet across. That's enormous for something you need to launch into space. No rocket has a payload fairing large enough to accommodate a solid mirror of that size. The solution was to make the mirror in segments, 18 hexagonal pieces that could fold up for launch and deploy once in space. Each segment is about 4.5 feet across, made of beryllium, coated in a microscopically thin layer of gold. Beryllium because it's light and strong and holds its shape even at extreme temperatures. Gold because it reflects infrared light exceptionally well. But making a segmented mirror that works as a single optical surface is incredibly difficult. Each segment has to align with its neighbors to within nanometers. That's billionths of a meter. Tolerances so tight that a human hair would look like a massive object by comparison. Get the alignment wrong by even a tiny amount, and the images blur. The telescope becomes useless. So each segment sits on a set of actuators, small motors that can adjust its position and tilt with extraordinary precision. 7 actuators per segment. 126 actuators total, all working together to create a perfectly aligned mirror surface. The mirror had to fold for launch. The segments on the sides swing inward, creating a narrower profile that fits inside the rocket fairing. Once in space, they swing back out and lock into position. The entire deployment sequence was designed to be automatic, controlled by pre-programmed commands and mechanical releases. Because human intervention wouldn't be possible. JWST operates at a location called L2, Lagrange Point 2, about 1 million miles from Earth. That's 4 times farther than the Moon. At that distance, repair missions like the ones that fixed Hubble's mirror problems in the 1990s are impossible. The telescope had to work perfectly the first time. Beyond the mirror, JWST needed a sunshield. The telescope observes in infrared wavelengths, which means it's detecting heat radiation from distant objects. But if the telescope itself is warm, it emits infrared radiation that swamps the faint signals from planets and galaxies. The solution is to cool the telescope down to incredibly cold temperatures. about -400 degrees Fahrenheit. Cold enough that the telescope's own heat doesn't interfere with observations. Achieving that temperature in space requires blocking heat from the sunday. The sunshield does this. It's a structure about the size of a tennis court, made of 5 layers of material thinner than a human hair. Each layer is coated to reflect sunlight and radiate heat. The layers are separated by gaps so that heat can dissipate without conducting from one layer to the next. The sun-facing side of the sunshield gets hot, reaching temperatures around 200 degrees Fahrenheit. The side facing the telescope stays incredibly cold. The temperature difference across the thickness of the sunshield is over 600 degrees. The sunshield also had to fold for launch. The 5 layers had to compress down into a package small enough to fit in the rocket, then deploy in space with each layer separating and tensioning properly. 147 release mechanisms, 292 pulleys, 90 tensioning cables, thousands of individual parts that all had to work in sequence, in the vacuum of space, at temperatures the hardware had never experienced during testing on Earth. If any single mechanism failed, if a cable snagged or a release didn't trigger, the sunshield wouldn't deploy properly and the mission would be over. The rocket carrying JWST lifted off from French Guiana on Christmas Day 2021. The Ariane 5 rocket, one of the most reliable launch vehicles in the world, but still a controlled explosion, carrying $10 billion of irreplaceable hardware. The ascent took about 26 minutes during which engineers and scientists around the world watched telemetry screens and held their breath. Any failure during launch would mean the loss of the telescope and 3 decades of work. The rocket performed perfectly. It delivered JWST to the correct trajectory, heading toward L2. Over 2 weeks, JWST unfolded itself in space, step by step, mechanism by mechanism. The telescope transformed from a compact package into its operational configuration. First, the solar array deployed, providing power. Then the high-gain antenna unfolded, establishing communication with Earth. The sunshield pallets extended, unfolding the collapsed layers and beginning the process of separation. Each membrane had to unfurl, had to tension properly, had to lock into position. Controllers on Earth sent commands and waited for confirmation that each step had worked. If something went wrong, there were backup procedures, alternative sequences, ways to try to recover, but everyone knew that some failures would be unrecoverable. The sunshield deployed successfully, all 5 layers separated and tensioned. The temperature differential across the shield began to establish itself. Then the mirror segments began to unfold. The wings on each side of the primary mirror swung out from their launch positions. Latches locked them in place. The secondary mirror, which reflects light collected by the primary mirror into the instruments, deployed on its support structure. More latches, more confirmations. Then came the fine alignment. Each of the 18 primary mirror segments adjusted its position using those actuators. Controllers uploaded commands that moved segments by tiny amounts, fractions of a millimeter, adjusting tilt and position. They used starlight to check alignment, pointing the telescope at bright stars and examining how the image looked. Initially, each mirror segment created its own image of the star, 18 separate dots of light. The goal was to align all 18 segments so perfectly that those dots merged into a single, sharp point. The process took weeks. Small adjustments, observations, more adjustments. Gradually, the 18 images moved closer together. They overlapped, they merged, and finally, JWST achieved perfect alignment. The segmented mirror was working as a single optical surface. The image quality exceeded expectations. The telescope was ready. First light came in July 2022. NASA released the first images publicly, and the world saw what JWST could do. Galaxies from the early universe appeared in unprecedented detail. Their light stretched into infrared wavelengths by cosmic expansion. Nebulae where stars are being born revealed structures that previous telescopes could only hint at. The depth of field, the clarity, the sheer amount of detail in every image demonstrated that the 3 decades of work had been worth it. But JWST wasn't just built to take pretty pictures of distant galaxies. One of its primary science goals was characterizing exoplanet atmospheres, finding out what gases exist on planets orbiting other stars, searching for biosignatures. The instruments include spectrographs specifically designed for this purpose, capable of splitting infrared light into thousands of individual wavelengths and measuring the brightness of each one with extreme precision. K2-18b was identified as a priority target early in the mission. The planet had been discovered years before JWST launched, detected by its transits across its star. Ground-based telescopes and earlier space telescopes had measured its size, mass, and orbital period. They confirmed it was in the habitable zone, but characterizing its atmosphere required JWST's sensitivity, required the ability to detect the tiny absorption signals from atmospheric molecules during transits. The first observations happened in 2023. JWST pointed at K2-18b's star and waited for the planet to transit. During the transit, which lasts a few hours, the telescope collected as much light as possible. The spectrograph split that light into component wavelengths. Detectors recorded the spectrum. Data streamed back to Earth where scientists began analysis. The initial results showed water vapor clearly. Not surprising for a world that's expected to be an ocean planet, but confirmation is always important in science. Methane appeared in the spectrum as well. And then, in detailed analysis of the absorption features, the dimethyl sulfide signature emerged, faint but present. The wavelengths where DMS should absorb light were showing reduced brightness compared to the baseline spectrum. One observation isn't enough for a definitive detection. Instruments can have errors, calibration can be off. Random noise in the data can create features that look like real signals but aren't. So JWST observed K2-18b again during a later transit. And again, each observation added more data. The telescope continues operating. Every 33 days, K2-18b transits its star, and JWST has the opportunity to observe. Not every transit is observed because the telescope has other targets, other science programs that need time. But regular observations continue, building up more data, refining measurements of atmospheric composition. Scientists are looking for other molecules now, searching for additional biosignatures that might confirm or challenge the interpretation that life exists on K2-18b. The capability that JWST has given us is remarkable when you think about it. We can point this telescope at a star 120 light-years away, wait for a planet to pass in front of that star, and read the chemical composition of that planet's atmosphere by analyzing starlight that has filtered through a few hundred miles of gas. This represents the culmination of everything humans have learned about light, optics, materials science, aerospace engineering, and planetary science. Building JWST required solving problems that didn't have obvious solutions, creating materials that could survive launch and deployment, designing mechanisms that would work in the vacuum and temperature extremes of space, writing software that could control alignment to nanometer precision, training teams of people to work together on a project that would take longer than many of their careers. And it almost didn't happen. Multiple times during development, the project faced cancellation. Budgets grew beyond original estimates. Technical problems emerged that took years to solve. Schedules slipped repeatedly. Critics called it a waste of money, questioned whether the science justified the cost, argued that the resources could be better spent on other programs. The project survived because enough people believed in what it could accomplish, believed that the ability to characterize exoplanet atmospheres, to search for biosignatures, to potentially answer whether life exists beyond Earth was worth the investment. The detection of dimethyl sulfide on K2-18b validates that belief. This is exactly what JWST was built to do. Finding biosignatures, detecting the chemical traces of potential life on distant worlds, opening a new era in the search for life beyond Earth. Before JWST, we could speculate about whether ocean worlds might host life. Now we can actually look and see what's in their atmospheres. We can gather evidence. We can make measurements. We can do science instead of just theorizing. Other planets are being observed. JWST is characterizing atmospheres on dozens of exoplanets, building a catalogue of what different types of worlds look like chemically. Rocky planets, gas giants, ice giants, super-Earths, mini-Neptunes. Each one teaches us something about how planets form, what conditions exist on their surfaces, whether they might be habitable. K2-18b is one discovery among many, but it's potentially the most significant because of that dimethyl sulfide detection. The telescope will continue operating for years. Its fuel supply limits how long it can maintain its position at L2 and how often it can adjust its pointing. Current estimates suggest at least a decade of operation, possibly longer if fuel consumption is lower than expected. During that time, it will observe thousands of objects, more exoplanet atmospheres, more distant galaxies, more star-forming regions. Each observation adds to humanity's understanding of the universe. But the K2-18b observations will remain special. This might be the telescope that first detected life beyond Earth, the instrument that answered one of humanity's oldest questions by finding the chemical signature of biological activity on another world. That's the kind of discovery that changes how we see ourselves, how we understand our place in the cosmos, what we think is possible in the universe. A species that learned to fly just over a century ago can now detect molecules on planets orbiting other stars. We went from the Wright Flyer to JWST in roughly one human lifetime, from wooden biplanes that barely got off the ground to a space telescope that can read atmospheric composition across interstellar distances. That trajectory of progress, that acceleration of capability, is one of the most remarkable things about human civilization. And we're not done. Future telescopes are being designed that will be even more capable than JWST. Larger mirrors, more sensitive instruments, broader wavelength coverage. The technology continues advancing. Each new generation of telescopes reveals things the previous generation couldn't see. JWST showed us dimethyl sulfide on K2-18b. The next generation might show us oxygen, ozone, chlorophyll signatures, or other biosignatures that would strengthen the case for life even further. The search for life beyond Earth has moved from philosophy to observational science. We have the tools now. We have the methods. We have telescopes capable of detecting biosignatures if they exist. K2-18b is probably just the first of many discoveries. More Hycean worlds will be found. More atmospheric observations will be made. More potential biosignatures will be detected. The catalogue of possibly living worlds will grow, and with each new entry, our understanding of how common life is in the universe will improve. JWST made this possible. This telescope, this incredible machine floating 1,000,000 miles from Earth, collecting infrared photons that have traveled for decades or centuries across space, splitting them into spectra, and revealing the chemistry of distant worlds. It's one of humanity's greatest achievements, a tool that lets us touch, in an informational sense, places we'll never physically visit. A bridge across the void, built from metal and gold and human ingenuity. The light from K2-18b keeps arriving. Every second, new photons that left that world in 1904 complete their journey and reach Earth. Some hit JWST's mirror and contribute to observations. Others pass by, continuing into deep space, carrying their information forever into the expanding universe. The stream of light never stops. As long as K2-18b's star shines, as long as the planet continues orbiting and transiting from our perspective, light filtered through its atmosphere will arrive here, carrying chemical signatures we can read. And somewhere in that atmosphere, if the biosignature interpretation is correct, organisms are releasing dimethyl sulfide right now, living their lives, metabolizing, reproducing, evolving, completely unaware that 120 light-years away, a species they'll never meet has built a telescope and detected their waste products. We found them without them knowing we exist. We're watching their chemistry from across the galaxy. That's the power of astronomy. That's what JWST has given us. The ability to detect life we can never visit, on worlds we can never touch, using nothing but starlight as our probe. On Earth, tiny creatures called phytoplankton drift in the ocean. Single-celled organisms so small, you need a microscope to see them, but there are trillions upon trillions of them floating in the upper layers of the ocean, using sunlight to convert carbon dioxide and nutrients into energy through photosynthesis. They're the foundation of the marine food chain. They produce much of the oxygen in Earth's atmosphere. And as a byproduct of their metabolism, as they process sulfur compounds from seawater, they release dimethyl sulfide into the air. Its waste, biological exhaust, the chemical signature of microscopic life doing what it does to survive. On K2-18b, 120 light-years away, something is releasing the same gas into a very different atmosphere. The question that keeps scientists awake at night is simple but profound. What exactly is producing that dimethyl sulfide? Is it life following the same metabolic pathways as Earth's phytoplankton? Is it something entirely different that happens to create the same molecule through different chemistry? Or is there some non-biological process we haven't discovered that can generate dimethyl sulfide without any living organisms being involved? The biological explanation is the most straightforward. Life on K2-18b could be metabolizing sulfur compounds, just like phytoplankton do on Earth. The chemistry of sulfur metabolism is well understood here. Organisms take in sulfate or other oxidized forms of sulfur from the environment. They use enzymes to reduce that sulfur, stripping away oxygen atoms and incorporating the sulfur into various biological molecules. During this process, some of the sulfur ends up in dimethyl sulfide, a volatile compound that escapes into the atmosphere. The process isn't particularly complex. It doesn't require advanced biochemistry or specialized cellular machinery. Simply organisms can do it. If life exists on K2-18b and follows similar metabolic rules to life on Earth, then dimethyl sulfide production makes sense. The planet has an ocean covering its entire surface. That ocean would contain dissolved sulfur compounds, just like Earth's oceans do. Sulfur is a common element, found throughout the universe, incorporated into rocks and minerals and dissolved in water wherever liquid water exists. Any organism living in K2-18b's ocean and processing nutrients from that water would encounter sulfur, and if their metabolism includes pathways for sulfur reduction, dimethyl sulfide would be a natural byproduct. The organisms themselves could be microscopic. They don't need to be large or complex to produce the quantities of dimethyl sulfide that JWST is detecting. On Earth, individual phytoplankton cells are tiny, but their collective impact is enormous. Trillions of cells, spread across millions of square miles of ocean, each one releasing tiny amounts of dimethyl sulfide, add up to detectable concentrations in the atmosphere. K2-18b could host similar populations. Vast numbers of single-celled organisms floating near the ocean's surface where starlight penetrates, using whatever form of photosynthesis or chemosynthesis works in their environment, incidentally producing dimethyl sulfide as they metabolize. The environment they'd be living in would feel alien by Earth standards. That hydrogen-rich atmosphere pressing down on the ocean's surface, the stronger gravity making buoyancy and movement different from what we experience. The reddish light from the red dwarf star providing energy at wavelengths shifted compared to our yellow Sunday. But none of these differences necessarily prevent life. They just mean that life there would be adapted to local conditions in ways that might look strange to us. Photosynthesis on K2-18b would work differently than on Earth if it exists at all. Our photosynthesis uses specific wavelengths of visible light, primarily red and blue portions of the spectrum. The chlorophyll molecules in plant cells and phytoplankton are tuned to absorb those wavelengths efficiently. K2-18b's star emits more infrared and red light, less blue and ultraviolet. Organisms using photosynthesis, there would need different pigments, molecules tuned to capture the wavelengths that are actually available. The biochemistry would be adapted to what the star provides. Or perhaps photosynthesis isn't the dominant energy source on K2-18b. Perhaps life there relies more heavily on chemical energy from the ocean itself. On Earth, deep-sea hydrothermal vents support entire ecosystems that don't depend on sunlight. Microbes extract energy from chemicals dissolved in hot water flowing from the vents. Hydrogen sulfide, methane, iron compounds, other reduced chemicals that react with seawater and release energy. Those microbes form the base of food webs that include larger organisms, all living in complete darkness, all powered by chemistry rather than light. K2-18b could have similar systems. The planet's interior would be hot from the combination of gravitational compression and radioactive decay of elements in its core. That heat has to go somewhere. It likely drives volcanic activity and creates hydrothermal vents on the ocean floor, just like Earth has. Those vents would release mineral-rich hot water into the cold ocean. The chemical gradients created where hot vent water mixes with cold ocean water represent potential energy sources that life could tap into. Organisms could evolve to extract energy from those gradients, to feed on the chemicals flowing from the planet's interior. These chemosynthetic organisms could produce dimethyl sulfide as well. The sulfur metabolism that creates DMS doesn't require photosynthesis. It's a separate pathway that organisms can use regardless of their primary energy source. Whether an organism gets its energy from light or from chemicals, if it's processing sulfur as part of its metabolism, dimethyl sulfide can result. So even if K2-18b's life is primarily chemosynthetic rather than photosynthetic, the DMS signature would still make sense. The combination of both energy sources is also possible. Surface waters where starlight penetrates could host photosynthetic organisms. Deeper waters and the ocean floor could host chemosynthetic communities around hydrothermal vents. The two systems could coexist, each exploiting different niches in the environment, together creating a complex biosphere that produces multiple gases we can detect from Earth. The methane in K2-18b's atmosphere could come from chemosynthetic microbes. The dimethyl sulfide could come from photosynthetic plankton near the surface. Different organisms, different metabolic strategies, all contributing to the atmospheric composition. But we need to be careful about assuming too much. Everything described so far is based on Earth as the only example of life we know. We're extrapolating from one data point. Life on K2-18b could be fundamentally different in ways we haven't imagined. The basic chemistry might be the same because chemistry is universal, but the implementation could be radically different. Different genetic molecules instead of DNA, different amino acids building different proteins, different cellular structures, different reproductive strategies, different everything except the underlying chemical reactions that happen to produce dimethyl sulfide. For life, there might not be cellular at all. On Earth, all life is based on cells, compartmentalized units, with membranes separating inside from outside. That's the only way we know life to work. But it's possible that life could organize differently. Maybe on K2-18b, living systems don't have cell membranes. Maybe they exist as chemical networks in the water itself, self-organizing metabolic processes that reproduce and evolve without discrete physical boundaries. We don't know if such systems are possible, but we can't rule them out. Our understanding of what life can be is limited by our experience, with only one form of it. The timescale for life to develop on K2-18b is comparable to Earth's. The planet is a few billion years old, orbiting a star that formed in the earlier generations of the galaxy. Life appeared on Earth within roughly the first billion years after the planet became habitable. The exact timing is uncertain because the early fossil record is sparse and ambiguous, but definitely within the first billion years and possibly much faster. If K2-18b's conditions became suitable for life early in its history, organisms could have had 2 or 3 billion years to evolve and diversify. That's a long time. Long enough for simple single-celled organisms to potentially evolve into more complex multicellular forms, though we have no evidence yet of anything beyond microbial life. The dimethyl sulfide signature is consistent with simple organisms, plankton-like creatures floating in the ocean. Whether evolution on K2-18b has produced anything more complex, anything like the animals and plants we see on Earth, we can't tell from current observations. Complexity doesn't necessarily show up in atmospheric chemistry. A planet could have complex ecosystems and still have an atmosphere that looks like it's dominated by microbial processes. The alternative explanation to life is that some non-biological chemistry is producing dimethyl sulfide on K2-18b. Scientists have searched hard for such processes. They've modeled chemical reactions that could occur in the ocean or atmosphere. They've looked at what happens when sulfur compounds interact with water, hydrogen, starlight, minerals, heat, pressure. They've examined geological processes that might release sulfur gases. So far, no one has found a plausible abiotic pathway that creates dimethyl sulfide in the quantities being detected. That doesn't mean such a pathway doesn't exist. It means we haven't discovered it yet. The universe is full of chemistry we don't fully understand, reactions we haven't observed, conditions we haven't recreated in laboratories. K2-18b might be doing something novel, some chemical process that doesn't occur on Earth because the conditions are different. The hydrogen-rich atmosphere, the high pressure, the specific mix of minerals dissolved in the ocean, the intensity and spectrum of starlight, all of these factors could combine in ways that create dimethyl sulfide without biology. If that's the case, if there's an abiotic explanation, then the dimethyl sulfide on K2-18b wouldn't indicate life. It would just be interesting chemistry, a reminder that planets can produce complex molecules through purely physical and chemical processes. We'd be disappointed because finding life is more exciting than finding novel chemistry. But we'd also learn something valuable about what planets can do, about the range of chemical environments that exist in the universe, about the need to be careful when interpreting biosignatures. The scientific process requires holding both possibilities in mind. The data shows dimethyl sulfide is present. The most likely explanation, based on Earth's example, is biological production. But the less likely abiotic explanation can't be completely ruled out until we understand K2-18b's chemistry much better than we currently do. Additional observations will help. Finding other biosignatures would strengthen the biological interpretation. Detecting oxygen or phosphine or other gases that on Earth are associated with life would make the case more compelling. Conversely, if detailed modeling finds an abiotic pathway that can produce the observed amounts of dimethyl sulfide under K2-18b's conditions, that would weaken the biological interpretation. Science follows the evidence wherever it leads. Right now, the evidence points toward life as the most parsimonious explanation. But that could change as we learn more. The organisms producing dimethyl sulfide on K2-18b, if they exist, are living their lives completely unaware of Earth. They don't know we exist. They don't know we've detected them. They're simply doing what living things do, metabolizing nutrients, reproducing, competing for resources, evolving in response to their environment. The dimethyl sulfide they produce is just waste, an accidental byproduct of staying alive. They're not trying to signal us. They're not attempting communication. They're just living, and their living happens to create a chemical signature we can detect from 120 light-years away. If there's intelligent life on K2-18b, if organisms there have evolved complex brains and developed technology and turned instruments toward the stars, they'd be facing the same communication problem we are. The 120-year light delay makes conversation impossible. Send a signal today, and the response arrives in 240 years. Multiple human lifetimes. Civilizations rise and fall in that span. Whatever question you asked would be irrelevant by the time the answer came back. Whatever answers they sent would address questions from a civilization that no longer exists in the form it did when it asked. So we're isolated, not by impossibility of detection, but by the tyranny of distance and the speed of light. We can know they're there, we can study their atmosphere and learn about their chemistry, but we can't meet them, can't visit, can't exchange ideas in any meaningful time frame. The universe has filled itself with life, if K2-18b is any indication, and then spaced those islands of biology so far apart that they remain forever separate, connected only by the thin thread of information carried in starlight. Yet there's something profound in even that limited connection. Before these observations, K2-18b was just a planet, a data point in a catalogue. Mass, radius, orbital period, distance. Numbers on a chart. Now it's potentially a living world. A place where organisms exist, where metabolic processes create and destroy molecules, where evolution shapes populations over time. The dimethyl sulfide detection transforms how we see that distant point of light. It's not just a planet anymore. It might be a biosphere, a world as complex and dynamic as Earth in its own way. And if K2-18b hosts life, then we're likely not alone in the galaxy. The Milky Way contains hundreds of billions of stars. A significant fraction of those stars have planets. Many of those planets sit in habitable zones. If life arose on K2-18b, if it emerged independently of Earth, then the conditions that allow life must be common rather than rare. The universe creates life wherever it can. Chemistry becomes biology, given the right ingredients and enough time. We're part of a pattern, not an exception. That realization changes everything. Earth isn't the living world. It's one of potentially billions of living worlds, each following its own evolutionary trajectory, each hosting organisms adapted to local conditions, each producing its own unique atmospheric signature. Some might have oxygen-rich atmospheres like ours. Others might be hydrogen-dominated like K2-18b. Some might be methane worlds, carbon dioxide worlds, ammonia worlds. The diversity could be enormous, limited only by which chemical combinations allow stable liquid solvents and metabolic processes. Life under a hydrogen sky, organisms swimming in endless oceans beneath an atmosphere we couldn't breathe, under light from a star cooler and dimmer than our sun, on a world where gravity pulls 3 times harder than Earth, metabolizing sulfur and releasing dimethyl sulfide into the air, reproducing, competing, evolving. Completely unaware that 120 light-years away, a species called humans has detected their existence and is trying to understand what they are. The symmetry is beautiful in a melancholic way. We're both alive, we both exist in the same universe, subject to the same physical laws, made of the same elements, forged in ancient stars. We're both complex enough to process energy and information, to maintain ourselves against entropy, to create order in small pockets of space-time. We're both products of billions of years of cosmic evolution. Chemistry that became biology that became ecosystems. We're fundamentally similar in that way, but we're separated by a gulf so vast that connection beyond detection is impossible. We'll never compare notes on what life is like on our respective worlds, never share what we've learned about the universe, never acknowledge each other's existence in any direct way. The hydrogen sky of K2-18b and the nitrogen sky of Earth will never meet. The organisms beneath those skies will never meet. We're alone together, each world an island in an ocean too wide to cross. Yet knowing they might be there matters. It changes how we see Earth. Our evolution isn't the only story of increasing complexity over billions of years. Our existence isn't unique. Just one example of what happens when a planet has the right conditions for the right amount of time. That's humbling and elevating simultaneously. Humbling because we're not special in the way we once thought. Elevating because we're part of something larger. A universe that consistently produces life, that seems almost eager to create biology wherever physics and chemistry allow. The dimethyl sulfide drifting through K2-18b's atmosphere right now is the same molecule that drifts through Earth's air above our oceans. Universal chemistry, universal biology, perhaps. Life using the same tools, the same reactions, the same metabolic pathways across interstellar distances. That suggests deep regularities in how life works, principles that transcend individual planets and emerge from the fundamental properties of atoms and molecules. Carbon forms 4 bonds, nitrogen forms 3, oxygen forms 2, hydrogen forms 1. These facts don't change from planet to planet. The chemistry built on these facts produces similar results wherever it operates. So K2-18b might host life that's alien in its details but familiar in its fundamentals. Different organisms, different ecosystems, different evolutionary history, but the same basic chemistry, The same kinds of molecules performing the same kinds of functions. Proteins made of amino acids catalyzing reactions, nucleic acids storing genetic information, lipids forming membranes, carbohydrates providing energy and structure. The toolkit of life might be universal because chemistry is universal. Or not. Maybe K2-18b's life uses completely different molecules. Maybe the apparent similarity in dimethyl sulfide production is superficial, the result of convergent chemistry rather than convergent biology. Maybe drilling down into the details would reveal two forms of life so different that comparing them is meaningless, except at the most abstract level of self-organizing chemical systems. We don't know. We can't know without closer study, and closer study isn't possible across 120 light-years with current technology. So we're left with questions. Beautiful, tantalizing questions that drive science forward. Is it life? What kind of life? How similar to or different from Earth's version? What does it look like? How does it reproduce? What's its evolutionary history? Has it developed complexity? Does it have ecosystems with energy flowing through food webs? Are there predators and prey, cooperation and competition? All the dynamics we see on Earth, or something entirely different? The answers will come slowly, if they come at all. More observations, better instruments, decades of patient study, gradually building a picture of K2-18b's chemistry detailed enough to constrain the possibilities. Maybe in 50 years, we'll have enough data to say definitively whether the dimethyl sulfide is biological. Maybe in 100 years, we'll have telescopes powerful enough to detect other biosignatures or rule them out. The process of science operates on long timescales when studying objects 120 light-years away. But the work continues. JWST keeps observing. Other telescopes add their data. Scientists analyze and model and debate. The catalogue of what we know about K2-18b grows entry by entry, and somewhere under that hydrogen sky, if the biosignature interpretation is correct, organisms keep producing dimethyl sulfide, keep living their lives, keep adding to the chemical signature we're detecting from Earth. The connection persists, fragile and one-way, but real, two worlds linked by photons carrying information across the void. The distance to K2-18b is 120 light-years. That number is easy to say, but almost impossible to truly comprehend. Light travels at 186,000 miles per second. Nothing in the universe moves faster. To understand what that distance means in practical terms, consider our fastest spacecraft. Voyager 1, launched in 1977, is currently the most distant human-made object from Earth. It's been traveling for nearly 50 years and has covered about 15 billion miles. That sounds impressive until you realize it's only about 0.2% of 1 light-year. Voyager 1 is moving at roughly 38,000 miles per hour. At that speed, reaching K2-18b would take approximately 2 million years. Even if we could travel at 1% of light speed, which is far beyond anything we can currently achieve, the journey would take 12,000 years. That's roughly the span of time from the end of the last ice age to the present. The entirety of human civilization would fit into the travel time. Generation after generation would be born, live, and die aboard the spacecraft. The mission would outlast empires. The people who arrived at K2-18b would have no cultural connection to the people who launched the mission. They'd barely remember Earth, except as a distant origin point in their history. At 10% of light speed, still wildly beyond our capabilities, the trip drops to 1,200 years. That's the time from Charlemagne to the present. From the early Middle Ages to modern times, still far too long for any individual to make the journey, still requiring generation ships or some form of suspended animation we haven't invented, still separating the launch generation from the arrival generation by dozens of lifespans. Even at light speed itself, which Einstein's relativity tells us is impossible for anything with mass to achieve, the journey takes 120 years. A person born the day the ship launched would be long dead before arrival. Their children would be dead. Their grandchildren would be old. Only their great-great-grandchildren would see K2-18b And that's the absolute best case. The physical limit imposed by the structure of space-time itself. You can't go faster than light. The universe doesn't allow it. So visiting K2-18b isn't just difficult. It's effectively impossible with any foreseeable technology. The distance is simply too vast. The timescales are too long. Human lifespans are too short. We can detect that world. We can study it from afar. We can know it exists. But we can't go there. We can't walk on its surface, which doesn't exist anyway because it's all ocean. We can't sample its water or study its organisms directly or confirm through close observation that life actually exists there. Communication faces the same barrier. The speed of light limits not just physical travel but information transfer as well. Radio waves travel at light speed. Send a message to K2-18b today and it arrives 120 years from now. If someone receives that message and responds immediately, you get the reply in another 120 years, 240 years round trip. The person who sent the original message is dead. Everyone they knew is dead. Several generations have passed. Whatever prompted the original message is ancient history by the time the response arrives. Imagine trying to have a conversation under those conditions. You ask a question, your great-great-great-grandchildren receive the answer. They ask a follow-up question. Their great-great-great-grandchildren receive that answer. Each exchange takes multiple human lifetimes. Continuity is impossible. Context is lost. The conversation becomes meaningless because the participants keep changing. You can't build understanding or relationship across gaps measured in centuries. This is the universe's cruelest reality. It allows life to arise on many worlds, potentially creates conditions for biology on planets scattered throughout the galaxy, produces organisms capable of wondering about their place in the cosmos. And then it spaces those worlds so far apart that they can never actually meet, never communicate in real time, never share what they've learned or acknowledge each other's existence in any direct, meaningful way. We're all trapped in our local neighborhoods, able to detect neighbors but unable to visit them. K2-18b and Earth exist simultaneously right now in this moment. Organisms on both worlds, if K2-18b has organisms, are metabolizing nutrients, reproducing, interacting with their environments. The two biospheres are happening in parallel, both products of billions of years of evolution, both complex systems of chemistry maintaining themselves against entropy. But we can never compare them side by side. We can never study one from the perspective of the other except through the delayed and limited information carried by light. The light we're seeing from K2-18b right now left that planet in 1904. If intelligent life exists there, and they're looking back at Earth with their own telescopes, they're seeing our world as it was in 1904, before radio, before airplanes became common, before the Industrial Revolution fully transformed the planet. They'd see a world with some technological development, but nothing like what exists now. No satellites. No artificial lights bright enough to detect from interstellar distances. No radio signals. Just a natural planet with biosignatures in the atmosphere indicating life, but no signs of technological civilization. We're seeing them the same way, K2-18b, as it was in 1904. If they've developed technology since then, if they've built cities or spacecraft or communication networks, we won't see evidence of that for another 120 years. The K2-18b of 2024, their present moment happening simultaneously with ours, is invisible to us. We can only see their past. They can only see ours. We're ships passing in the night, each seeing the other's wake but never the ship itself. This temporal displacement adds another layer of isolation to the spatial separation. Even the information we receive is outdated by more than a century. K2-18b could have changed dramatically since the photons we're analyzing left. The organisms producing dimethyl sulfide could have evolved in new directions. Environmental changes could have altered the atmospheric composition. Catastrophic events could have disrupted the ecosystem. We wouldn't know. We won't know until the light carrying that information completes its journey to Earth. Similarly, anything we learn about K2-18b tells us only about its past. The data we collect today describes a world from over a century ago. We're always playing catch-up, always looking backward, always separated from the present state of distant worlds by the light travel time. This is fundamental. This is unavoidable. This is how the universe works when distances are measured in light years. The isolation extends to every potentially living world we might discover, each one separated by light years, each one visible only in the past, each one unreachable except by information traveling at light speed. We can build a catalogue of life-bearing worlds, map where they are, characterize their atmospheres, estimate their ages and conditions, but we can never visit them, never interact with them in real time, never bridge the gaps that separate one biosphere from another. This creates a strange situation where we might be surrounded by life, by millions or billions of living worlds throughout the galaxy, and yet remain fundamentally alone. Alone in the sense that we can't reach out and touch the other living worlds, can't exchange ideas with them, can't learn from them except through passive observation of their atmospheric chemistry. The universe becomes a gallery of living worlds that we can look at but never enter. Paintings behind glass that we can study but never directly experience. Some people find this depressing. The idea that we might discover life is common but connection is impossible. That we're destined to remain isolated on Earth no matter how many other living worlds exist. That the gulf between inhabited planets is unbridgeable, not because we lack technology, but because physics itself forbids faster than light travel and communication. We're locked in place by the speed of light, confined to our local region of space by the sheer vastness of the distances involved. But there's another way to see it. The isolation makes each living world more precious, not less. If we can never visit K2-18b, if we can never see another biosphere up close, then Earth becomes even more valuable. This is the one place we can actually experience life directly, the one biosphere we can walk through, swim in, study in detail, protect or destroy. Every other living world is abstract, knowable only through spectral analysis and atmospheric modeling. Earth is real, immediate, ours in a way that nowhere else can ever be. The isolation also means that each biosphere evolves independently. K2-18b's organisms followed their own evolutionary path, shaped by local conditions, producing their own unique solutions to the problems of survival and reproduction. Earth's organisms did the same under different conditions. If we could somehow catalogue all the living worlds in the galaxy, we'd see the full range of what life can become. The entire space of evolutionary possibilities played out across billions of different environments. Each world is an independent experiment in biology, showing what happens when chemistry becomes life under specific conditions. That diversity has value. If we were all connected, if travel between inhabited worlds were easy, life might homogenize. Species from one world could spread to others. Ecosystems could mix. The unique character of each biosphere could be lost. The isolation preserves uniqueness. K2-18b will always have only K2-18b organisms. Earth will always have only Earth organisms. The separation maintains the independence that allows each world's evolution to continue along its own trajectory. There's a bittersweet beauty in knowing that other living worlds exist, but accepting that we'll never visit them. We're not alone in the universe. Life is out there, on worlds scattered through the galaxy, following the same basic chemistry we see on Earth, creating complex ecosystems from simple molecules and energy gradients. We can detect those worlds, we can learn about them, we can know they exist, but we can't go there. We can only observe from afar, watching ancient light and reading chemical signatures, forever separated by distances too vast to cross. K2-18b is 120 light-years away. The dimethyl sulfide we detected was produced in 1904. Whatever organisms created that gas are long dead now if they have lifespans anything like Earth's plankton. Their descendants are producing dimethyl sulfide in our present moment, but we won't detect that for another 120 years. The K2-18b of 2144. When those photons arrive, we'll be observing our world as it exists today, They'll see 21st century Earth 120 years from now if they're looking. The cycle continues indefinitely. Information flowing across the void at light speed, creating connections between worlds that can never physically touch. Each observation is a snapshot of the distant past. Each detection tells us what was happening there when our great-great-grandparents were young. We're looking backward through time, seeing other worlds as they were, never as they are. And they're looking at us the same way. If they're looking, seeing our past, our history, the version of Earth that existed before anyone alive today was born. This is the nature of interstellar distances. This is what it means to be separated by light years. Not just space, but time. Not just distance, but delay. Everything is old by the time we see it. Everything is already different from what the light shows us. We're studying history, not current events, learning about the past of distant worlds while their present remains invisible and inaccessible. The philosophical implications are profound. We exist simultaneously with potential life on K2-18b, sharing the universe in the same moment. But we can never share information about that moment. By the time they know what Earth is like today, we'll be experiencing our future. By the time we know what K2-18b is like today, they'll be experiencing their future. We're out of sync, running parallel but offset by the light travel time, forever unable to align our knowledge with the present reality of each other's worlds. Does it matter? Does the isolation diminish the significance of finding life elsewhere? Some would say no. The detection of dimethyl sulfide on K2-18b, if it indicates life, is one of the most important discoveries in human history, regardless of whether we can ever visit. Knowing we're not alone changes our perspective on existence. It transforms Earth from the only living world to one of many. It suggests that life is a natural outcome of cosmic evolution, not a miraculous exception. That shift in understanding is valuable independent of any practical ability to interact with other living worlds. Others might argue that discovery without interaction is incomplete. That knowing life exists on K2-18b, but being unable to study it closely, unable to see the organisms or understand their biology in detail, leaves us with frustrating partial knowledge. We can detect chemical signatures, but can't answer deeper questions about how that life works. What it looks like, how it evolved. We're limited to remote sensing of atmospheres, learning about alien biospheres through the narrowest of keyholes. Both perspectives have merit. The isolation is real and limiting. We won't get complete answers about K2-18b's potential life from Earth-based observations alone. But incomplete answers are still answers. Partial knowledge is still knowledge. Knowing that dimethyl sulfide is present, that something is producing it, that the most likely explanation is biological, tells us something profound about the universe, even if it doesn't tell us everything about K2-18b specifically. The search continues despite the limitations. We observe more transits. We look for additional biosignatures. We study K2-18b's atmosphere in ever greater detail. Each new observation adds a piece to the puzzle. Each detection constrains the possibilities. Slowly, over years and decades, we build a picture of what that distant world is like. Not a complete picture, not the kind of detailed understanding we have of Earth, but enough to answer some questions, enough to know whether life is likely there, enough to understand in broad strokes what kind of world it is. And we look at other planets. K2-18b isn't the only potentially habitable world within range of our telescopes. Thousands of exoplanets have been discovered. Hundreds sit in habitable zones around their stars. Many are candidates for atmospheric characterization. JWST and future telescopes will observe them, searching for biosignatures, cataloging which worlds show signs of life and which don't. We're building a census of living worlds, or at least potentially living worlds, mapping the distribution of life across our galactic neighborhood. Each discovery adds to our understanding. If we find dimethyl sulfide on multiple ocean worlds, that suggests the biological explanation is correct. If we find it only on K2-18b, that might indicate something unusual about that specific planet. If we find many different biosignatures, on many different worlds, that tells us life is common and diverse. If biosignatures are rare, that tells us something different about how often life emerges. The pattern matters as much as individual detections. But every world we study suffers from the same limitation: distance. All of them are light years away. All observations show us the past. All potential life is unreachable. We're mapping a universe of living worlds we can never visit, cataloging biospheres we can never walk through, building knowledge of alien life we can never meet. The project is fundamentally one of remote observation. We're astronomers studying biology across interstellar distances, using starlight as our only tool. The distance to K2-18b will never change. 120 light-years is fixed. The planet isn't moving significantly closer or farther from Earth over human timescales. We're locked in this relationship, observing from afar, learning what we can from photons that take over a century to arrive. That's the reality. That's what the universe gives us. Not connection but detection. Not interaction but observation. Not meeting but knowing. It's less than we might hope for, but more than we had before. Telescopes powerful enough to characterize exoplanet atmospheres existed, so we accept the limitations and work within them. We observe. We analyze. We learn. We build understanding piece by piece, from the fragments of information carried by ancient light, and we marvel at what we can accomplish despite the constraints. A species confined to one planet can still detect molecules on worlds 120 light-years away, can still search for life across interstellar distances, can still reach out, in an informational sense, and touch places that physical travel will never allow us to visit. That's something. That's remarkable. That's what makes us human, this drive to explore and understand even when direct exploration is impossible. Continue, Baden Wuschafen. K2-18b remains distant, unreachable, isolated from Earth by a void too wide to cross. But we know it's there. We know something on that world is producing dimethyl sulfide. We know the potential implications. The distance doesn't change those facts. The isolation doesn't diminish their significance. We found them, even though we can never meet them, and somehow, knowing that makes the universe feel both emptier and fuller at the same time. Emptier, because the gaps between living worlds are so vast. Fuller, because those worlds exist at all, scattered like islands in the cosmic ocean, each one potentially hosting its own version of the miracle we call life. So here we are, somewhere out there, 120 light-years away, on a world covered in ocean beneath a hydrogen sky. Something might be alive, releasing gases, processing chemistry, existing, completely unaware that here, on a small blue planet circling a yellow star, other living things have noticed them. We found each other across the impossible distance. We detected their chemistry through the analysis of ancient starlight, and we will never meet, never communicate, never share what we've learned about the universe or acknowledge each other's existence in any direct way. Is that tragic? Or is it something else? The question sits at the heart of what the K2-18b discovery means. We've potentially found life beyond Earth, confirmed that we're not alone in the universe, and simultaneously confirmed that we are alone, in the sense that each living world remains isolated in its own region of space. Connection and separation happening at the same time. Knowledge and limitation intertwined. The answer to, are we alone, appearing to be both no and yes, depending on how you define alone. What we can choose is how we respond to this reality. One response is despair. We found potential neighbors and can never visit them. We discovered we're not alone and simultaneously discovered that our loneliness is permanent and unbridgeable. Every living world is a prison defined by light years. Every biosphere evolves in isolation with no possibility of contact with other biospheres. The universe filled itself with life and then made sure none of that life could reach any other life. That's depressing if you focus on it, but there's another response, another way to interpret the same facts. The isolation makes each living world irreplaceable. K2-18b, if it has life, is the only place in the universe with K2-18b organisms. Nothing exactly like them exists anywhere else. Their evolutionary history is unique. That uniqueness has value. If we could travel easily between living worlds, if distance weren't a barrier, ecosystems might homogenize. Earth species could spread to other planets. Alien species could arrive here. The distinct character of each biosphere could blur. The isolation preserves that distinctness. K2-18b will only ever have K2-18b life. Earth will only ever have Earth life. Each world remains its own evolutionary experiment, showing what life becomes under its specific conditions. The universe maintains a diversity of biospheres that wouldn't exist if living worlds could mix. Think about what that means for Earth specifically. This planet, this biosphere, this collection of organisms and ecosystems and evolutionary lineages is one of a kind. Yes, K2-18b might have life. Yes, other worlds might be biologically active, but none of them are this. None of them have redwood forests or coral reefs or elephants or whales or the specific trillion-member ecosystem of microbes in your gut. All of that is exclusively terrestrial. It evolved here. It exists here. It will never exist anywhere else unless we deliberately transport it, which we can't do across light years. That makes Earth precious in a way that transcends whether we're alone. Even if life is common, even if billions of planets host biospheres, Earth is still the only source of Earth-type life, the only place where you can see oak trees, or touch a cat, or swim with dolphins, the only place where human consciousness emerged from evolutionary processes and started asking questions about its place in the cosmos. Other worlds might have their own forms of consciousness, their own organisms wondering about meaning and existence, but they're not this consciousness, not this specific way of being aware and alive. The K2-18b discovery, if it holds up under continued scrutiny, shifts how we think about both commonality and uniqueness. Life might be common. The conditions that allow chemistry to become biology might arise on many worlds throughout the galaxy. But each instance of life is simultaneously unique, shaped by local conditions, following its own evolutionary trajectory. We're part of a pattern and an exception at the same time. Common in the sense that life emerges repeatedly, unique in the sense that Earth life is unrepeatable. This duality resolves the old question about whether we're special. We are and we aren't. We aren't special in that life probably exists on many worlds. The universe creates biology wherever it can. K2-18b suggests we're not a miracle, not a one-time accident, not the only place where molecules organized into self-replicating systems. But we are special in that this specific collection of organisms, this specific evolutionary history, this specific biosphere exists nowhere else. Every living world is special in that sense. Every instance of life is both an example of a common pattern and a unique, unrepeatable expression of that pattern. The dimethyl sulfide on K2-18b tells us that life uses similar chemistry across interstellar distances. The same molecules, the same metabolic pathways, the same waste products. That suggests deep regularities in how biology works, principles that emerge from the properties of atoms and elements. Carbon forms 4 bonds everywhere. Water is a polar solvent everywhere. These facts don't change from planet to planet. The chemistry built on these facts produces similar results wherever it operates. But similar chemistry doesn't mean identical organisms. Earth's phytoplankton and whatever produces dimethyl sulfide on K2-18b might use the same metabolic pathway but could look completely different, have different cellular structures, different reproductive strategies, different ecological roles. The underlying chemistry converges. The implementation diverges. Life finds similar solutions to similar chemical problems, but expresses those solutions through different biological forms shaped by local evolutionary pressures. This is what we're learning about life through astronomy. Not just that it might exist elsewhere, but what kind of phenomenon it is. Life is chemistry that crosses a threshold into self-organization and reproduction. That threshold can be crossed in many places, But what emerges on the other side of that threshold is shaped by environment, by available energy sources, by planetary conditions, by random mutations, and selection pressures operating over millions or billions of years. Universal in principle, diverse in practice. The same basic phenomenon producing countless variations. K2-18b gives us one more data point, one more example of what life can be. If the dimethyl sulfide is biological, we're seeing life adapted to high gravity, hydrogen atmospheres, ocean worlds, red dwarf starlight. That's different from Earth's conditions. The adaptations would be different. The organisms would be different. But the fundamental nature of being alive, of maintaining metabolic processes and reproducing and evolving, that's the same. Different expressions of the same underlying pattern. And we're part of that pattern. Earth life isn't separate from or different than the pattern. We are the pattern expressing itself through terrestrial conditions. K2-18b, life, if it exists, is the pattern expressing itself through Hycean world conditions. Other worlds with other conditions will produce other expressions. The universe is showing us that life is something it does, a regular feature of cosmic evolution, as natural as stars and planets and galaxies. Chemistry becomes biology when conditions allow. That happens repeatedly. We're one outcome. K2-18b might be another. The philosophical implications are profound. For most of human history, we thought life was unique to Earth. We were special, chosen, fundamentally different from the rest of the universe. Then we discovered we're on a planet, orbiting a star in a galaxy among billions of galaxies. Not central, not unique in location, but maybe still unique in hosting life. The K2-18b discovery, along with other potential biosignature detections, is removing that last claim to uniqueness. We're not unique in hosting life either. We're part of a pattern, one instance of something that happens throughout the universe. That's humbling. It removes humans from the center of cosmic significance. We're not the point of the universe. We're not the reason everything exists. We're an outcome of natural processes, one of many such outcomes, important to ourselves but not particularly important to the cosmos at large. The universe would continue fine without us. It contains countless other worlds, some of them potentially hosting life, all of them following physical laws that don't care about human existence. But it's also elevating in a way. If life is common, if the universe consistently produces biology, then being alive connects us to something larger than our individual existence, or even Earth's biosphere. We're part of a universal phenomenon, part of what the universe does. The atoms in our bodies were forged in stars. The chemistry that makes us alive is the same chemistry operating on distant worlds. We're made of the universe, and we're the way the universe becomes conscious of itself. That's true whether we're the only instance or one of many. Being common doesn't make it less meaningful. It makes it more meaningful because it shows life is a fundamental feature of reality rather than a bizarre exception. The K2-18b discovery invites us to reconceptualize what being alive means. We're not isolated accidents. We're expected outcomes of physical processes. Given a planet in the habitable zone with liquid water and available energy and enough time, life emerges. It has on Earth. It might have on K2-18b. It probably has on countless other worlds. The universe wants, in an impersonal, physical sense, to create life. The laws of physics and chemistry make it almost inevitable given the right conditions. We're the result of that inevitability playing out on one particular planet. That changes the weight of existence. We carry forward a process that's billions of years old and operates throughout the universe. Every living thing on Earth, from bacteria to blue whales, is part of that process. Every organism is chemistry that became biology, that became evolution, that became the specific forms we see around us. And on K2-18b, if life exists there, the same process produced different forms adapted to different conditions. The process is universal. The outcomes are diverse. We're one outcome among many. The light from K2-18b continues arriving every second. Photons that left 120 years ago are completing their journey. They carry information about a world as it existed in 1904. We capture some of those photons with telescopes. We analyze the information they carry. We learn about that distant planet's atmosphere and chemistry. And in 2144, if we're still here and still observing, we'll capture photons leaving K2-18b today. We'll see their present as our future. The cycle continues, information flowing across the void at light speed, connecting worlds that cannot physically touch. Right now, at this moment, you're on Earth. This planet, this specific world with its nitrogen-oxygen atmosphere and liquid water oceans, and millions of species shaped by billions of years of evolution. You're part of Earth's biosphere. You're made of atoms that were once in other organisms, that will eventually be in future organisms, that cycle through the ecosystem in patterns as old as life itself. You're connected to everything alive on this planet through deep time and shared chemistry and common ancestry, stretching back to whatever first organism started the whole process. And somewhere, 120 light-years away, if the biosignature interpretation is correct, organisms are living their own version of existence. Different chemistry in details, different forms, different evolutionary history, but the same fundamental phenomenon. Life maintaining itself against entropy, chemistry organizing into complexity, metabolism and reproduction and adaptation. The pattern expressing itself through different conditions but recognizable as the same pattern nonetheless. You'll never meet them, never see what they look like or understand how they live, but you know they might be there. You know that Earth's biosphere might be one of many. You know that being alive connects you not just to other Earth organisms, but to a universal process that operates throughout the cosmos. You're part of something larger than you realized, part of what the universe does when chemistry becomes biology. That knowledge sits quietly in your mind now. Earth is one living world among potentially many. You are one living organism among trillions on this world and potentially countless more on other worlds. Unique and common simultaneously. Special and ordinary at once. Important to yourself and to those around you, but cosmically unremarkable, except as one more instance of life's pattern. And maybe that's the right perspective. Not the center of everything, but part of everything. Not alone, but isolated. Not the only life, but the only Earth life. Connected to the cosmos through the atoms in your body and the chemistry keeping you alive. Aware that on distant worlds, other organisms might be living their own versions of existence, following their own paths through the space of biological possibilities, creating their own ecosystems and evolutionary stories. The universe filled itself with life, scattered it across galaxies on countless worlds, made sure each world's life stayed separate, evolved independently, remained unique, created diversity through isolation. Connected everything through common chemistry and physics while keeping actual living things apart through the tyranny of distance and the speed of light. The dimethyl sulfide drifts through K2-18b's atmosphere right now. Molecules produced by whatever organisms live in those endless oceans beneath that hydrogen sky. The same molecule drifts through Earth's atmosphere, produced by phytoplankton in our seas. Universal chemistry, parallel biology. Separate worlds doing similar things in different locations, never meeting but knowable to each other through the information carried by light. That's the truth K2-18b reveals if its dimethyl sulfide truly indicates life. We're part of something larger, something the universe does consistently, something that makes chemistry become complexity and complexity become consciousness. We're one instance of that something, one expression of life's pattern. Unique in details, but part of a universal phenomenon. And here you are, on Earth, alive, part of it all, unique and common, special and ordinary, connected and alone. That's what it means to be human in a universe where life might be everywhere, but everywhere is too far away to visit. That's the perspective the K2-18b discovery offers. Not despair at the distance, not frustration at the isolation, but acceptance of what is, and wonder at what might be, and gratitude for the one living world we can actually touch and experience and call home. And that's enough. That's everything. That's what we are.